research was based on the established functions of cardiac cell-derived extracellular vesicle, microRNAs, and myocardial infarction. My name is Dilara, and my mentor was Dr. David Lundy at the Medical University of Taipei. So about me before we start, I'm a rising senior that attends an Islamic school. I'm in New Jersey, and my hobbies are to plant or draw in my free time. So what are we exactly observing in this research, right? So we want to know what extracellular vesicles specifically are. And what those are secreted by cells and contain cargo such as proteins and microRNAs. And microRNAs are an important element in gene expression because they have great potential for medical treatment. A population of extracellular vesicles could contain hundreds of different unique microRNAs. And these microRNAs could have multiple possible target genes. And in our previous work in the lab, a population of the cells from the human heart were studied, right? And extracellular vesicles were isolated in vitro functional tests that showed that they were cardioprotective. So our data set revealed the presence of microRNAs, including microRNA 21.5p, microRNA 590, microRNA 199A, and etc. So when a person intakes medical treatment after a heart attack, we want to know what's specifically in the treatment, which is kind of the point in our research, because we want to know what's in our treatments after we intake medicine after we have a heart attack. This involves researching a list of database human microRNAs associated with these activities. In the future, we hope to gain a better understanding of how cells interact and the human heart more by technology advancing. So what is myocardial infarction? Over the course of years, your arteries eventually begin to build up plaque until it unexpectedly ruptures, and that causes a myocardial infarction known as a heart attack. Let me see. So cell therapy employs tiny fragments of our cells to fix issues that could develop into diseases. At the University of Miami Miller School of Medicine and the University of Maryland School of Medicine, researchers have discovered that stem cells from a person who was not related and could be a complete stranger could help restore a patient's own cells after damage was done to the heart. So patients who have tried everything or have no treatment at all now have hope because of this discovery. A person who was born with serious illness caused by a single malfunctioning gene, this issue could be fixed by injecting a a functioning copy from someone of the gene into the cell. It's also possible to deliver the new gene into the place where it's needed in the body. So long non-coding RNAs are these special molecules that have been recently related to their control of heart cell development and repair. And these long non-coding RNAs have the ability to interact with microRNAs, which can change the way they behave. Okay. So for our project, I was provided with a data set of microRNAs in extracellular vesicles in the human heart by my mentor, Professor Lundy. I researched into the list of microRNAs to determine whether it was connected to myocardial infarction, and then I would take the microRNAs I took, and I looked into the data to learn about the targets and functions of those microRNAs. So research has revealed that many things produce microRNAs, like the microRNA 143p, where HSA also stands for human microRNA, which means microRNA. So the MIR means miRNA, and HSA stands for human, by the way. And some microRNAs affect things like the muscle growth and heart health. They could even help reduce scarring in tissues. So we could think of them as tiny helpers in the body. So in our study, as you can see here, this was the table from our lab that was studied. And I would take these microRNAs and I would research each one to see if they're related to myocardial infarction. So in the study, we looked at microRNAs related to heart tissues. And we focused on human miRNAs with the HSA at the start. And we found that micro 215P is the most common, and then micro 202.5P and so on. So this list goes from to the least common. So HSA micro 215P is important and studied a lot. It helps fix arteries using tiny parts from cells called exosomes, and these parts clean up cells, kind of like a human cell vacuum. This micro 215P makes this happen by reducing a protein called CYP1L2. And this could protect the heart more. So diving into the diversity of human microRNAs, the study showed that micro 590 and micro 199A have been found to improve cardiomyocyte proliferation and assist in cardiac repair. And microRNA treatment after myocardial infarction in mice led to serious regeneration in the heart and almost full recovery of cardiac functional indices. So researchers examined if delivering microRNAs directly could enhance cardiomyocyte growth and heart regeneration. If microRNAs could stimulate cardiomyocyte proliferation and cardiac generation, it would lead to improved cardiac function. So this investigated the impact of microRNAs on heart recovery and function post-heart attack. Results showed that these microRNAs indeed brought benefits, enhancing performance and recovery of the injured heart. So microRNAs are tiny parts that help build tissues and cells. They can also talk to other cells. Researchers used a special method to find microRNAs supporting blood vessel and cell growth. And this discovery could lead to better heart disease treatments by promoting heart cell growth, improving cardiac function, and reducing damage. Okay, so interaction between microRNAs and extracellular vesicles. So extracellular vesicles are a small structure, and microRNAs communicate with other organisms in the heart, so they're an intercellular communicators. They both have an influence on the functions of genes and cell activity. So heart issues are a major issue, just like blood channel blockages that harm the body. For recovery of the heart after damage, researchers are trying to employ extracellulars and microRNAs. So the reason that they were chosen is because they play a role of communicating with one another and are messengers. So they're also known to transport information between other cells. So since they have the ability to communicate, they could grow new heart blood vessels, which is beneficial for regeneration of the heart and so much more after a heart attack. So microRNA profiling in heart health, small cell components known as EVs play a crucial role in cardiac conditions like heart failure, which is the main focus of our study. So when these specific microRNAs are combined, the diagnosis of heart attacks could be made even more accurately. So a study of the human microRNA known as microRNA 590 and microRNA 199A have been discovered to improve cardiomyocyte proliferation and help in heart repair, like we said. We looked at how treating mice with microRNAs after a heart attack could impact their recovery. And what we found was actually quite remarkable. The mice showed quick and great heart regeneration and almost a complete recovery after a heart attack. And one microRNA that was important in this was microRNA 21, which has been associated with heart issues like heart attacks and heart failure. So our study aims to explore whether directly introducing certain microRNAs could boost the growth of heart cells. This in turn could potentially enhance overall heart function by reducing the extent of injury caused by a heart attack. So when heart cells are exposed to palmitate, known as a fatty acid, we've observed a connection between micro-21 and cell death. While we're still uncovering the specifics, it seems that micro-21 plays a role in controlling the process of cell death, which we could also call apoptosis. So cell death, also we could call as apoptosis, occurs after a heart attack occurs. So this molecule is a primary target of microRNA 210-3P. When the levels of EF and A3 dropped within the extracellular vesicles, we observed a decline in the extracellular vesicles' ability to aid in heart recovery after a heart attack. So, yeah. Okay. With further research into various human microRNA types, some haven't still been fully researched. They don't go into detail because it hasn't been fully studied yet. And this actually happened when I was researching some microRNAs, and I asked my professor, and I said, this doesn't go into specific depth, which was microRNA 155 5P. And he said that it hasn't been fully discovered yet. So that's a problem in researching this. So this microRNA could serve as an example of this to comprehend it better. This microRNA is related. So while this microRNA is related to coronary artery disease, which is known to be a huge risk for heart attack, it doesn't fully go into detail on its direct impact on heart attacks. So during heart attacks, micro 155 5P rises, affecting apoptosis related proteins. So cells with the microRNA 155 suppressed by AMO 155 survive longer due to reduced apoptosis, thanks to microRNA 155's impact on their intercellular communication. So treating cardiac diseases. So recent studies suggest that manipulating microRNAs could be a therapeutic avenue for cardiac diseases. Specifically after a heart attack, microRNA 29 influences collagen and fibrosis patterns in cells. This insight opens new possibilities for addressing heart attacks and cardiac dysfunction. So in this study, we focused on to completely understand what's in our medical treatment after a myocardial infarction. So microRNAs have great potential in medical treatment research. They can be useful in detecting heart disorders. Like the microRNA 133A is known to be a useful way to diagnose diseases and to see how bad the situation has gotten. So to predict the outcome of these diseases, researchers are looking at several microRNAs altogether. So we've also covered how extracellular vesicles have gained attention in the medical field due to their significant potential. However, there are some issues to this. A study investigated that EVs and microRNAs in myocardial damage post-heart attack. They isolated heart cells to understand EV impact on injury. The findings revealed that AMI EVs caused cell death, implying their potential role in cardiac damage. Despite advancements in microRNA development methods, understanding their behavior across various heart disease stages remains a challenge still today. So the complexity of microRNAs' effects on cells, genes hinders their direct use as treatments. So our conclusion is, just like genetic controllers, microRNAs might become future medicines. Researchers are actively investigating microRNA biology and its connection to extracellular vesicles. So researches could revolutionize and make us better understand of heart diseases and start more personalized treatments with advancing technology. So enhancements are being explored, including safe delivery of, microRNAs treatments to the heart. We could have techniques like nanoparticle-based delivery and viral vectors that are being examined for accurate microRNA transfer to heart cells. So with continuous progress, personalized medicine could become a reality, tailoring to treatments to patients based on genetic profiles, illness severity, and treatment response. Yeah, thank you.